Hello guys, welcome to another video. In this video we're going to learn about the PCR, that means polymerase chain reaction. So polymerase chain reaction or PCR is a method of making unlimited copies of DNA using a test tube, a heat source, and a few simple reagents such as template, primers, DNA polymerase, deoxyribonucleoside, triphosphate, and buffer. And the history of PCR is the enzyme was first isolated and characterized from extracts of E. coli bacteria by Arthur Kornberg in 1957. Then Carey Mullis considered the possibility of duplicating cell DNA under in vitro condition. In 1960. Then in 1983, Carey Mullis synthesized this existing information into one of the most powerful tools of molecular biology called the polymerase chain reaction, PCR, or in vitro cloning. That is, PCR is also known as in vitro cloning. In technical terms, PCR may be defined as a rapid procedure for in vitro enzymatic applications of specific DNA sequences using two oligonucleotide primers that hybridize to opposite strands and flank the region of interest in the target DNA. In this video we are going to learn about the PCR in details. So let's get started. What are the applications of polymerase chain reaction? Here we can see some general application of PCR, that is diagnosis of monogenic diseases, diagnosis of mutation diseases, DNA typing, evolutionary trends and disease susceptibility studies, PCR and forensic science, detection of human infectious diseases, detection of rash oncogenes, PCR and DNA vaccine production, etc. So these are the general applications of polymerase chain reaction. Then here are some research application of polymerase chain reaction. What are the research applications? Such as direct sequencing of in vitro amplified DNA, engineered DNA to meet specific needs, detection of mutation, detection of gene expression, specific amplification of DNA species, geometric amplification of unknown DNA sequence through inverse PCR, analysis of DNA sequences in individual gametes, evolutionary analysis, etc. So these are the research applications of PCR. Then here are some reagents and equipments for PCR, such as optimization of PCR, primer selection, buffer, DNA polymerase, deoxyribonucleotide, triphosphate, cycling parameters, etc. These are the reagents or equipments for PCR. Then here are some PCR components, such as Taq polymerase, DNA sample, primers, nucleotides, mix, buffer, PCR tube, thermal cycler, etc. These are the components of PCR. Here are the basic protocol of PCR. First one is optimize magnesium chloride concentration. Prepare a 10x amplification buffer containing magnesium chloride at 10-fold the optimal concentration. Then test the optimized amplification buffer by preparing a reaction cocktail for a specific for a single PCR. If PCR is successful, prepare more reaction cocktail for storage at -20 degrees Celsius. Then add 68 microliter 15 microgram per milliliter template DNA and 9 microliter of H2O. Then add 1 microliter 2.5 microliter tag DNA polymerase. Overall, the reaction mixture with 100 microliter mineral oil to prevent evaporation. Heat samples for 90 seconds at 94 degrees centigrade to denature the DNA. Incubates at 55 degrees centigrade for 2 minutes to anneal. Incubate the sample at 72 degrees centigrade for 3 minutes to extend. Repeat the step 7 to 9 for another 29 cycles. Electrophoresis 10 microliter from each sample on an agarose non-denaturing polyacrylamide gel. Stain the gels with ethidium bromide. Examine the stained gel. So these are the basic protocol of PCR. Here we can see the PCR process is separated into 4 steps. That means this basic protocol is separated into 4 steps. First one is initialization, second one is denaturation, and third one is annealing, and last one is elongation. So initialization temperature 94 to 96 degrees centigrade, and the time duration is 2 to 10 minutes. To activate DNA polymerase and denatures contaminants in the mixture. In the case of colony, PCR screening the steps also facilitates cell lysis to release DNA material and denatures other cellular proteins. So this is the first step or initialization step, and here we can see the first step and initialization step. Then second step is denaturation. The double stranded DNA are broken by heating the reaction 94 to 98 degrees centigrade for 20 to 30 seconds. Here we can see that double stranded DNA are broken by heating. Then next step is annealing. After DNA strands separated, primers bind to a complementary sequence in the DNA template to guide DNA polymerase replication. In the step, the reaction temperature is lowered to 50 to 65 degrees centigrade for 20 to 40 seconds to allow for optimal primer for annealing. Here you can see that primer annealing occurs in this step. Then primer attached to the template. Then last step is elongation. At the end of the last cycle, there is a final elongation step that keep the reaction mixture at 72 to 78 degrees centigrade for 5 to 15 minutes. This ensures that any remaining single-stranded DNA is fully extended after the last PCR cycle. Final holding step keep PCR at 4 to 15 degrees centigrade for an indefinite time, keeping the products for short-term storage. Here you can see the elongation step. Elongation is occurs in 3' to 5' or 5' to 3' in this way. Then these are the steps of PCR. Then here we can see the tag DNA polymerase, and the most common DNA polymerase enzyme used in PCR is tag polymerase. Tag polymerase are thermostable DNA polymerase that allows multiple cycles of amplification without needing a new enzyme addition. Tag polymerase discovered by Dr. Kary-Mullis in 1980s. Then there are some attributes that can be utilized for specific PCR applications, and the attributes are proofreading abilities, routine PCR, high specificity, whole blood PCR, high yield, high fidelity, difficult templates for long range PCR, etc. So these are the attributes for PCR application. Then visualize the DNA fragments. To visualize the DNA fragments after amplifications by PCR, gel electrophoresis is used by comparing the gel bands to a molecular weight marker. Researchers can estimate the size of the amplified product to know if their desired genes were successfully amplified. Here we can see visualize the DNA fragments in 350 base pair. Then next one is important factors in PCR. There are 5 important factors such as nucleotide composition of DNA template, DNA polymerase enzyme choice, buffer components, primer design, additives and inhibitors, etc. So let's get started. First step. The first factor is nucleotide composition of DNA template. We know that GC-rich template have more hydrogen bonds compared to AT-rich templates. This results in incomplete strand separation, which may cause the PCR fail. In addition, high GC content template tend to lead to more secondary structures which can arrest the polymerase, leading to premature termination. In this cases, secondary structures destabilizers such as DMSO or dimethyl sulfoxide can be added. At the same time, AT-rich template also required special attention because of their low primer annealing temperatures. Non-specific primer annealing can occur under low annealing temperature, and primer specificity can retained using a combination of lowered extension temperatures that uses of additives such as TMAC, that is tetramethyl ammonium chloride. Then next, long templates are difficult to amplify because of their higher likelihood of DNA template being broken or degraded by depurination. Depurination refers to a chemical reaction during which the beta and glycosidic bond in the purine nucleoside is cleaved to release a nucleotide base due to hydrolysis. As Taq DNA polymerases will not extend through a purinic position during replication. Then depurination is an important limiting factor of long template amplification. This can be avoided by using a higher PC reaction buffer and decreasing the denaturation time to limit depurination, or by using proofreading DNA polymerase. So this is all about the nucleotide composition of DNA template. Then next factor is DNA polymerase choice. As mentioned earlier, there are many variations of DNA polymerase commercially available, such as most common is DNA Taq polymerase is most common. The next factor is buffer components, each with different features depending on the DNA template. Choosing the correct DNA polymerase and pairing it with an appropriate buffer can be critical for successful reaction. Then next factor is primer design. Apart from DNA polymerase choice, having a good pair of primers can also dictate the outcome of the PCR. Some consideration to keep in mind include the length of the primer, the annealing temperatures, and the sequence of the primer. For example, the pair of primers should not have complementary sequences, otherwise they can easily lead to self-dimer or primer-dimer formation and complete with template primer annealing. So this is about primer design. And next one is additive and inhibitors. Many naturally occurring substances such as polysaccharides, tannic acid, EDTA, or ethylene diamine tetraacetic acid can also inhibit PCR. Some inhibitors may degrade or modify the DNA template, while others can disrupt the annealing of primers to DNA or alter the DNA polymerase activity. Inhibitors can be with sample specific nucleic acid isolation protocol, or sometimes the use of additives can help in reducing the inhibition. For a complete list of PCR additives and their mechanism of action, please refer to our knowledge base. A combination of favorable factors contributes to a successful PCR. There are some additives such as ammonium sulfate, TMAC, formamide, betaine, DMSO, EcoSSB, T4-gene, deoxyguanosine, etc. Then there are some PCR inhibitors such as protein fats, humic acids, phytic acids, immunoglobulin G, bile, calcium chloride, EDTA, heparin, ferric chloride, etc. So that's all about the factors, and this is all about the PCR. That's all for today. See you in the next video. Thanks for watching.